Hello, my name is Victoria, and I will continue on with how human gene therapy works and the processes in more detail. With the broad understanding of what gene therapy is, now we discuss its sciences. The need for human gene therapy comes from the genetic diseases found in today's world. With human gene therapy, we are able to treat the cause of the diseases and not just the effects. It is something that people are born with rather than obtain, such as other common diseases. Genetic diseases come from defective genes within the chromosomes, which are passed on hereditarily. With gene therapy, we can target the defective genes themselves and either restore or alter them to achieve the appropriate proteins that they have so lacked or restrict them as needed. As the science behind this study grows, it is important that gene therapy is to be used strictly for restoration and not beautification, as the tampering with genetic makeup has its moral and ethical concerns. The goal is to bring the patients to a standard form of health in which they are able to live without the aid of medical intervention. The most common vector, which is the transportation of the restorative gene to the cell, is adeno-associated virus, also known as AAV. AAV has been the most utilized vector for gene therapy for its biological makeup, allowing for the most possible treatment, as it can be easily modified and treated to fit the needs of some diseases. The genetic virus itself allows for modification as certain DNA pieces can be integrated into it and used to deliver to the host. Another reason this virus has been utilized is that it is mildly immunogenic. Because of this, it is able to signal for an immune response from the body enough for it to take it where it needs to go, still be safe enough to pass through to get where it needs to be in the cells. Lastly, when it goes into manufacturing these genes, AAV is able to be adjusted for the 4 different needs of the disease. These are the cells or area that is being targeted, the body's response to the gene, the means of which the treatment is given, and its promoters. Once we have our vectors, now we have the process in which we administer the new and healthy gene. The 2 main means of administering the healthy gene are in vivo and ex vivo treatment. In vivo treatment is typically used when the genetic disease is caused by a specific defective gene in a specified area. In vivo compared to ex vivo treatment differentiate, as in vivo treatment is given by injection to a specific area of the body where the issue arises from. An example of this would be inherited retinal disease, which causes a loss of vision due to the genetic disorder in the retina. Luxturna is then prescribed, which will then be administered under the eye. The virus, our viral vector, will then enter and carry the new gene to its specific location to start the gene therapy process. Once there, the body will then treat the vector as it does any other, and the virus will find itself in the cells and be able to deliver the gene, which will then make up for the defective gene and create proteins needed for the body to function properly. Next, we have ex vivo treatment, which calls for more than just an injection and in some cases requires weeks in hospitalization. Ex vivo treatment is seen more when it comes to the various genetic blood diseases such as SCD, which causes an increase in abnormally shaped red blood cells, causing a lack of oxygen to be held. In this treatment, we see the new genes actually limit the production of a certain gene to try and reduce the number of abnormal cells being produced. Ex vivo treatment involves applying the new gene to stem cells outside of the body. First, blood is drawn from the patient and then goes through the process of separating out the stem cells so that the modified genes and DNA can be then added to them in a laboratory as needed. The cells are then considered transduced. Some cases will call for chemotherapy to remove the defective cells from the body before anything else can be done. Once that process is complete, the new modified cells are then reintroduced to the body, typically through an IV infusion. Gene therapy is still a relatively new science. As of now, there has been many successes and multiple accounts of positive results from receiving this treatment. There is a wide variety of genetic diseases which can now receive this treatment. Right now, AAV and LV, lentivirus, are the 2 primary viruses used. However, there is already research being conducted to utilize more viruses as potential vectors. With the many successful cases being reported, studies have gone into the expansion of this biotechnology overall. Some therapies, as of now, are only able to offer partial treatment for some diseases due to issues pertaining to the edited cells and may not be permanent, or there is a disconnect. The aim is to expand the diseases in which they can cure fully. While some diseases, such as the ones that deal with muscle and neurological factors, may not be as prominent in their advancements so soon due to many reasons, it is expected that they will also be curable with time.